First of all, thank you so much for inviting me to present my research work. Myself, Professor Asifullah Khan from PIAS, Pakistan Institute of Engineering and Applied Sciences. My topic is advancement and applications of deep convolutional neural networks in medical image analysis. A glimpse of PIAS. PIAS is situated in the capital city of Pakistan, Islamabad. Before going into the details of my specific research problem related to deep convolutional neural networks and its exploitation in cancer analysis, let me very briefly discuss artificial intelligence and its recent rise. We know that AI is becoming very popular in the fields of robotics, drone swarm, bioinformatics, medical image processing, and high energy physics. Specifically in robotics, Tesla robot Optimus. Optimus is a humanoid robot designed to work in industry. Boston Dynamics develops a series of highly mobile robots that can be used for transportation and military applications. Similarly, Leonardo is a hybrid of drone and robot technology that can even ride on a skateboard. It has very good control mechanism. And in medical sciences, Da Vinci surgical system, it offers integrated technologies for robot assisted surgery. Then we have the drone swarm technology, which is becoming very popular. And as far as AI algorithms are concerned, particle swarm optimization and deep reinforcement learning are mostly used in drone swarm technology. Drone swarm can be used for search and rescue operation, disaster management, and even defense-oriented applications. And as far as natural language processing is concerned, the large language models such as ChatGPT are becoming very famous and they have given a boost in the AI field. We know that ChatGPT is an AI-based large language model which generates text just like humans. It can converse with humans on a wide range of topics and has the ability to learn the context of the discussion. Now we know that letting the machine learn the context of a discussion is a very intelligent feature, and ChatGPT has been able to learn this feature. Now we coming to the main topic of my research based on deep convolutional neural networks. The outline is as such. First, we are going to very briefly describe what is machine learning and deep neural networks. Then we focus on convolutional neural networks called CNNs. And then the exploitation of CNN in histopathology-based lymphocyte analysis and cancer diagnosis. Okay, so basics of machine learning and deep learning. We know that deep learning is a subfield of machine learning, and machine learning is a subfield of artificial intelligence. Now, what is machine learning? Machine learning algorithm tries to learn through example. So we have two phases, the training phase and the test phase. In the training phase, we provide data, and through data, the machine learning model tries to learn that particular problem. Once it learns, we call it a learned model, and we use it in the test case on the unseen data. However, if the problems are complex, they demand higher level of learning, and there comes deep learning. So traditionally, if the problem is not complex, we can use traditional machine learning algorithm. But if the problem is complex and it's a big data problem, then we need to use deep learning algorithms. So what is deep learning? Deep neural networks are a type of machine learning that are more suitable for big data problems, that is complex problems. Due to its deep and hierarchical learning mechanism, they can often produce accurate results than the traditional machine learning techniques. Basics of deep learning. A deep neural network consists of a hierarchy of layers. Each layer transforms the input into more abstract representation for the output layer to make predictions. Such as seen in this particular diagram, we have an input image. Initially, basic features like edges and curves will be detected, then intermediate level features in the intermediate layers, and then high level features at the higher level layers. Okay, before going into the details of deep convolutional neural networks, let's see what are the general neural networks. Neural networks are considered a collection of simple mathematical units called artificial neurons, and they perform complex functions collectively. So collectively we have an input layer, a hidden layer, and an output layer. Now imagine that if we have 100 or 1000 of hidden layers in between the input and output layer, so how much complex the neural network would be. And the number of hidden layers basically dictate the depth of the deep neural network. It is being considered that when we increase the depth of the neural network, that is the number of hidden layers, its representation ability increases. Now we come to convolutional neural networks, CNNs. Very briefly, convolutional neural networks are feed-forward neural networks, and their ability to dynamically learn domain-specific representation is of the utmost importance, because there is no need of designing manual descriptor and exploits the spatial correlation in the image. That's why they are considered good for image processing and computer vision application. CNN has the ability to hierarchically learn representation, and that is lower layers they learn generic features, and higher layer they learn domain specific features. Again, pictorially, the working of deep convolutional neural networks, we see that we have input image, then we have the convolution layer, then pooling layer, again convolution layer and pooling layer. So convolution layer and pooling layer might be used alternatively, and in between we can have the activation functions such as ReLU or even the normalization function. And then at the end we flatten these layers into one-dimensional layers and make them fully connected. And then at the last of this stage we have the softmax layer, which basically performs the classification. So in a sense we have the feature engineering phase, okay, through which we learn different features like shown over here. Initially we might learn edges, curves, then object paths at intermediate level, and then even the objects, the higher level features. So this phase is called the feature engineering phase, and the last phase is called the object detection or classification phase. Okay, so the different architectural innovations, we have witnessed a lot of innovative ideas as far as the architectures of CNNs are concerned. There was a competition in order to make deep and further deep convolutional neural networks. Then an idea came of the concept that we must use multi-path processing in the deep convolutional neural network. Just like in roads we have multiple paths. So, and then again the bypass concept, that we might have bypass connection, skip connection, or residual learning connection in addition to the main information processing connection. Similarly, architectural innovation in CNN continued. Similarly, the idea of squeeze and excitation, which basically give importance to the feature maps and rank the feature maps before giving them to the next block or layers. So we see that the importance is computed over here, and then the feature maps are ranked over here and then given to the next block. We also gave an idea of a channel boosted CNN, whereby we thought that instead of just processing the original feature spaces in the backbone of the CNN, why not compute auxiliary channels, artificial channels through auxiliary learners, and these learners could be 1, 2, 3, 4, 5, and so on, depending upon the need. So once these auxiliary learners learn the different feature spaces, these feature spaces are then combined with the original feature spaces and given to the backbone of the rest of the CNN architecture. Channel boosted CNN, as discussed, is a new idea to further enhance the learning capability of deep CNN by generating boosting input channels. The combination of original and auxiliary channel boosts the discrimination capability of CNN. In order to study the taxonomy of the recent CNN architectures, we have been able to publish a survey paper with the title of A Survey of the Recent Architectures of Deep Convolutional Neural Networks. It is published in a very reputed journal, Artificial Intelligence Review by Springer Nature. And in last 2 years, it has been cited more than 2,200 times. Among these are more than 110 books and proceedings. Also, very recently, we have been able to publish another survey papers which is related to vision transformer and the CNN transformer based variant. So vision transformer are also becoming very popular. Similarly, deep convolutional neural networks are also becoming very popular. And then we have hybrids of these techniques which are called as CNN transformer variant. So both have their good features being learned through the hybrid vision transformer. Some applications of deep CNNs. Deep CNN have shown exemplary performance on several computer vision and image processing tasks such as image classification, object detection, and even segmentation. Now, the third part of the talk is the specific research topic that we wanted to discuss. It is related to lymphocyte analysis and cancer diagnosis. So cancer analysis, we have a number of measures to analyze cancer like lymphocyte detection, mitosis detection, and nuclear detection. We are going to focus on lymphocyte detection. Let's see what is lymphocyte and its detection. Lymphocyte analysis and cancer. Human immune system has an important role in cancer prevention. Tumor infiltrating lymphocyte, also called TILs, serve as a vital biomarkers in cancer patients due to their role in eliminating tumor cells. So these cells, TIL, try to kill the cancer cell. And thus detecting and quantifying TIL enhances precision in tailoring treatments such as immunotherapy and chemotherapy. So tumor infiltrating lymphocyte, TILs, they infiltrate in the tumor and try to kill the cancers as shown over here. They come through the blood, enter the tumor cells and try to kill those tumor cells. So the concentration of TIL is important for diagnosis and a further analysis of the cancerous tissue. In classical pathology procedure, histopathological slides are prepared by taking some tissue samples and staining them with specific stains and placing them on slides. These slides are later examined by the pathologist under light microscope. However, this procedure is very time consuming and may face the issue of subjectivity because these are whole slide images and may be of the size of 50,000 by 50,000 pixels. So very difficult for a pathologist to even analyze a few of these whole slide images in a single day. So that's why there is a need of automating this system. And we say that therefore digital pathology has helped in digitization of these histopathological images that can be processed using computer algorithms. So first we digitize these histopathological images through the scanner, okay, and then once they are digitized, then we analyze them through automatic algorithms, specifically AI-based algorithms. However, even understanding these digital images and analyzing them automatically through AI technique has some challenges. Why? Because the automated histopathological analysis for lymphocyte detection may face these below challenges like variation in lymphocyte morphology, unavailability of pathologist label dataset, that is the truth table, presence of artifacts, and there might be some staining issues, some cluster presence of the cells, and then even we have variations in staining and variations from different labs as well. So just to give you pictorial glimpses of the challenges, for example, weakly labeled data is provided, so difficult to perform the training task. In this case, the count annotations are just given, not the position of the cells, nor even the shapes of the cells are provided. In this case, we have just been provided with the position, the centroids of the cells, not the shape of the cell. However, in this case, we have the mask annotation, so the full information is available. Variations at tissue level, we see that stroma regions, they have different morphology. Sparse lymphocyte have different type of morphology, and artifacts have even different colors and different morphology. Similarly, if the lymphocytes are clustered, very difficult to detect these individual lymphocytes in a cluster. Again, heterogeneous lymphocyte morphology, even the single cell of lymphocyte have different shapes and sizes as shown over here. So this is also a challenge. And then multi-organ representation. For example, the histopathological images of breast cancer would be different than the colon cancer and even different than the prostate cancer. You see that the details are different. So very difficult for a single AI-based algorithm to tackle all of these different cancer types. Again, the challenge of class imbalance, like stroma region is heavy in number. The lymphocytes shown in red are less in number, and similar the artifact regions are difficult to handle. So overall, lymphocyte detection and analysis, regular areas shown over here, clustered area which are difficult to analyze and diagnose, and even the artifact have different shapes and structures, while single lymphocyte are easy to tackle. So how do we address these challenges in lymphocyte detection and analysis? To address the aforementioned challenges, there is a need for an efficient automated diagnostic system. In this regard, deep learning has shown worthy performance due to its ability to automatically learn complex nonlinear function from complex data set. And we have developed a novel boosted channel fusion based CNN. We call it BCF LYMP detector for lymphocyte detection in multi-cancer histopathology images. So what are the attributes of the proposed detection system? The proposed network initially selects candidate lymphocytic region at tissue level. So the first part is to select the candidate lymphocyte regions at tissue level and then detect the individual lymphocyte cells at cellular level. The proposed technique generates diverse boosted channels by utilizing the feature learning capability of different CNN architectures. And these different CNN architecture provides us with the different artificial channels. Once we have the original channel of information and the different artificial channels, then we fuse all these channels gradually and systematically using our own proposed feature fusion block. So for this purpose, we have reported a new adaptive fusion block uh, which is able to combine and select the most relevant lymphocyte specific feature from the generated enriched feature spaces. So we have generated enriched feature spaces and then we combine them in a good manner. The proposed fusion block utilizes the idea of attention to give the most contributing feature maps high weightage, whereas neglecting the redundant features. So pictorially, our proposed architecture is as such. We see that we have pyramid type of architectures used for auxiliary learner 1 and then auxiliary learner 2. So this is the feature extraction stage. We, in addition to the original feature spaces, we also generate auxiliary feature 1 and auxiliary feature 2 to these learners, and then we fuse these features and we see that the fusion is performed at different scales. So these are pyramid type of architectures. At different level we combine or fuse the features. However, this fusion is not simple addition or concatenation. For this, we have developed our own CNN architecture which systematically fuse these enriched feature spaces. Then we have the mask RCNN type of framework whereby we have the FPN feature proposal network, and then we have the region proposal stage, and then even the detection and the mask head. However, the novelty of our work comes in this initial part where we, in addition to the original feature spaces, we create artificial channels and exploit those channels at different level of scale. So the whole concept is given over here. We have input image, auxiliary learner 1, auxiliary learner 2. It has its own feature maps. The auxiliary learner has own feature map. They are combined with the original features and we get the boosted feature map. Once we get the boosted feature map, then they are given to the proposed feature fusion block, which is like such. It is a CNN architecture developed to systematically and gradually exploit these information-rich feature spaces. And this provides us with the deep boosted channels. Now, these deep boosted channels are provided to the backbone of the CNN architecture that follows, like for example, the FPN in Mascar CNN type of setup. So how do we perform this fusion? The details are given over here. So we have a one by one convolution, batch normalization, ReLU, 3 by 3 convolution, again batch normalization, ReLU, one by one convolution, batch normalization, ReLU, and then we have the channel attention mechanism in order to give weightage or rank the important channels. And once we have the channel importance in hand, then we try to have the spatial attention, which gives the importance of each particular object inside the image, inside the feature map. So we have the spatial attention as well as the channel attention. So this fusion block that we proposed is able to systematically and gradually exploit the different feature spaces that we created artificially as well as the original feature space. Now, coming to results, the developed network has demonstrated promising results. The F score is 0.93 on Listo dataset and 0.84 on NuClick dataset. Similarly, the precision and recall score are very reasonable. Just to give you some visual glimpses of the detection process. So the lymphocyte, you see the green boxes shows that the lymphocyte cells are detected correctly, even in different morphology based circumstances. Okay, different shown even in clustered image and in images which are very difficult to detect the lymphocyte. Well, thank you so much for listening, and special thanks to the organizers of this reputed conference. If you have some questions, please use my email address, asif@pias.edu.pk. Thank you so much.